Hi everyone, we will share about the role of medical laboratory technologies in diagnosis COVID-19. As we know, in 2020, a significant threat to public health emerged in Malaysia. It started December 2019 from Wuhan City, China, and it's still spreading globally since its discovery. In lab, appropriate laboratory testing as a part of the critical role of medical lab technologies is providing a contribution in diagnosing and managing of medically important viral human pathogen SARS-CoV-2, as known as COVID-19. COVID-19 is recognized as a public health emergency of international concern. Inappropriate laboratory diagnosis of SARS-CoV-2, together with the intervention such as social distancing, contact tracing, and hand hygiene, is playing the most important role to alleviate the virus spreading. As MLT, my friend will share about how the role as medical lab technologies in this pandemic.
Hi, my name is Raagavendeeran. Today I want to talk about COVID-19 sample collection, sample transportation, and how to discard the sample. COVID-19 sample can be obtained from two sites. One is a pharyngeal site, second is oropharyngeal site. Only use synthetic fibers with plastic attached with artificial swabs to collect the sample. Don't use calcium arginate swab to collect the sample because it can inhibit the PCR products and also inhibit the viruses. Don't use the swab attached with wooden because it can cause injuries. Can collect the swab from both nostrils, but not necessary to collect the specimen from both sides if the mini-tip is saturated with fluid from the first collections. The swab is dispersed into a liquid medium is known as VTM, viral transport medium. VTM is composed with buffers to control the pH, protein to stabilize the virus, and other substances to control the osmolality. VTM is a leading device for viruses for demonstrating survival rate of many viruses at ambient temperature. VTM also can transport many viruses such as SSV, influenza, H1N1, and RSV, and adenovirus. Keep sample at 2 to 8 Celsius. If delay in running samples or delay in shipping more than 72 hours, should keep sample in -70 or below 8 temperature, and sample should be discarded in the yellow plastic bag with seeds.
Testing the presence of COVID-19. Infection with the virus causing COVID-19 is confirmed by the presence of viral RNA detected by molecular testing. Usually, PCR detection of viral RNA does not necessarily mean that a person is infected and able to transmit the virus to another person. This is how to do reverse transcription PCR. The specimen should be transferred to lysis buffer under biosafety cabinet. The lysis buffer should contain a guanidine based inactivating agent as well as non-denaturing detergent. Indeed, the buffer include in common commercial extraction platform such as Biomeric do contain guanidine detergents are able to inactivate any viral coronavirus. Similarly, universal transport medium that include guanidinium salt is available from Merlin Biomedical. Because this test is a reverse transcription method, the saliva or swab used to collect added to lysis buffer to disinfect the specimen as well as to stop degradation of the COVID-19. The clinical specimen should not be heated to 56 degrees Celsius for 30 minutes, as evidence suggests that this process may also degrade the coronavirus RNA even as it inactivates viral coronavirus. For RTK antigen, in Malaysia, the use of RTK antigen is made priority in cases as sample that require urgent sample for a prompt patient management to be given, such as screening for emergency and semi-emergency procedure or surgical cases with probability of COVID-19 infection. Screening for broad index BID cases with high probability or high suspicion of COVID-19. When lab receive the specimen, put swab into extraction buffer tube. Stir them well in an angle position. Remove the swab while squeezing the sides of the tube to extract the liquid from the swab. Press the nozzle cap tightly onto the tube. Drop extracted specimen 3 drops in a specimen well read within 50 to 30 minutes by 2 persons advisably. Thank you.
Hi everyone, my name is Aaron, and today I would like to give some information about the medical laboratory technologies part in COVID-19 diagnosis. So what we do in diagnosis of the COVID-19 actually. So as we all know, many tests are done nowadays to identify whether a patient is actually a real patient for COVID-19 or not. And we also do a lot of tests such as screening and also confirmation tests as well. So today I would like to talk about two laboratories, which is microbiology lab and also virology lab. Now virology lab is a sub-lab from microbiology lab. So for COVID-19 in microbiology lab, we will be receiving various samples from the patients, such as respiratory samples and other samples, for example blood samples and also stool samples and so on. But the main sample, which is quite very high in virology factors, virulent factors, is actually respiratory samples, such as tracheal aspirate samples for tracheal aspirate CNS, and then tracheal aspirate for AFB direct smears and so on. So when receiving the samples, MLTs should be wearing full PPE because it is a personal protective equipment for MLTs where it needs to be worn by everyone in the lab actually. But for this particular case, which is COVID-19 diagnosis, it is very important, pretty important. And after receiving the samples, we need to process those samples, and still, as usual, the person who is in charge of the sample still need to wear the PPE, fully worn PPE. So the samples are processed according to the SOPs given, and then later on the samples are released after validating the results. But mostly MLT's parts are till over here, where we just run the samples, process the sample, and then we just release the results. Validation is done by medical officers or scientific officers. And then the last part in microbiology lab, after receiving and processing the samples and also releasing the results, we need to dispose the samples with extra care taken, because it is a COVID-19 positive patient sample. So when disposing the samples, MLTs in charge should be in full PPE, as we all know from the start, where we receive the samples and then we process and then we release the results, and then till the last part, where MLTs need to dispose the samples, and then the samples sometimes are kept for 1 week period, and then for extra tests to be done lah. So the samples should be kept in a safe place, and in a place which where the samples won't be in a leaking position or something like that. Lastly, the samples would be autoclaved and then disposed. Okay, for the next part, where I would like to explain about what happens in virology lab and the role of MLT over there. Virology lab, as we all know, it is a sub lab from microbiology lab. So this is quite a major lab for COVID-19 diagnosis actually. So for the first part in virology, screening tests are ordered frequently by the doctors. Screening test is actually a rapid COVID-19 rapid test. It could be antigen or it could be in antibody type, but the one we usually use is antigen. So which means COVID-19 antigen test, COVID-19 rapid antigen test. Rapid tests are processed within a short period of time, with releasing the results in a short period of time as well. So normally the turnaround time for rapid test is actually 2 hours. So in this period, the samples would be received by an MLT in charge, and then the samples are processed by an MLT as well, and then the validation as usual goes to the scientific officer or the medical officers, and then the confirmation test is actually another test which is done by the virology lab, only virology lab. Rapid test could be done anywhere by medical laboratory technologists, but it is just a screening test and not a confirmation test. But for the main part, the PCR test, which is the confirmation test, MLT's important role in virology is processing the main sample, which is also a confirmation test called COVID-19 PCR test. PCR stands for polymerase chain reaction. So as usual, when receiving the samples, MLTs are in full PPE until the test is released with its result as usual. But we have few steps for this PCR test, which is, first step is RNA extraction, where we extract RNA from the samples. But this sample we receive in a viral medium, where the nasal swab or an oropharyngeal swab is sent in a viral medium, and then these processes would be carried out. The first step is RNA extraction from the sample, and then the second is reaction mixture is prepared for the sample. The third one is application process in a PCR machine, which is where the COVID-19 virus is detected, whether it's positive or negative. But this test could be taking up to 6 hours. So if a doctor wants to report the sample in a short period of time, they need to do the screening test only. But to confirm whether it's correct or not, to double check, you need to send for PCR test. So that's all the role of an MLT in virology and microbiology lab. Thank you.
Assalamualaikum and hi, my name is Nurshafiqah binti Mohd Sabri. So one of the crucial role of medical laboratory technologists that involve directly in the research and development department, they are working hard to produce a reliable and convenient rapid test kit for COVID-19. The role of medical laboratory technologists is mainly one of the team that works with doctors, professors and scientists that involved with the research. Why rapid test kit is important? As COVID-19 has been announced as a pandemic, thus with the existence of D614G strain of COVID-19, which is the super spreader, enormous of samples have been sent to the laboratory. Using the PCR method, there are many steps and time consumed to do the test. With the aid of rapid test kit, the time consumed can be lessened as well as the lives of many, because treatment can be started earlier. In May 2020, Ministry of Health has announced that antibody rapid test kit cannot be used as a confirmatory test for COVID-19. Although it aids in faster result, also the time consumed, there is limitation in the use of the kit. Why? Because presence of antibodies cannot confirm active infection. It might be that the patient has been infected before and the body has already producing the antibodies and the virus is not there anymore. And then the next one is that negative does not guarantee patient is not infected with COVID-19. Why? Because the person might just get infected with COVID-19 that the body does not produce antibodies yet. So maybe the COVID-19 virus is still in the body. Alright, and then the use of sample for the antibody rapid test kit is finger prick blood, which is not convenient for virus detection. And then it is recommended for the antibody rapid test kit to be used in the surveillance studies and epidemiological studies. In September 2020, a group of Malaysian scientists successfully produced the first rapid test kit that can detect COVID-19 as accurate as this performed in the lab. The innovation was led by Associate Professor Dr. Wanwar Datul Amani Wan Salim. The kit can detect COVID-19 using saliva. It is called Hybrid Rapid Test Kit for SARS-CoV-2. And then the test required for the confirmatory test for now shall be from antigen-based real-time reverse transcription, polymerase chain reaction test or antigen rapid test kit using nasopharyngeal swab for sampling. That's all from me. Thank you.
Okay, the conclusion, as we know, COVID-19 caused by SARS-CoV-2 has remained globally health emergency of an international concern, posing serious health, economic and social crisis. The early detection and characterization of the viral RNA from the various clinical samples such as upper respiratory tract, lower respiratory tract, blood, urine and stool, using standard, conventional and novel imaging technique will generate a great role in timely and rapid containing of the virus. The detection of anti-SARS-CoV-2 RNA using serological method at various weeks after the onset of illness will aid the clinical and epidemiological investigation of the virus, especially under the condition when room temperature PCR is negative for highly suspicious patients. However, information is required to validate accuracy and reliability of this portable immunoassay. During pandemic like the current threat of COVID-19, the role of medical lab technology is irreplaceable. The establishing countermeasure to unlock this era so that ensuring a valid, reliable and highly impactful result will be top critical priority of laboratory medical lab technologies always play a crucial behind the scenes role even after this crisis has passed. That's all from Group 5. Thank you.